So today we will talk about stem cell, the blood stem cell, and the types of blood cells that is going to develop into. Now, keep in your mind that stem cell is actually pluripotential type, and it gets developed into several types of blood cells. Okay, we were saying that the types of bloods that we're gonna get from the stem cell is firstly red blood cell, the granulocytes. This is a type of leukocytes, basophil, eosinophil, neutrophil, and we have a lymphocytes right here, and this lymphocytes is a granulocyte type. Okay, and we will get a monocyte that later will get develops into macrophage. We will talk about each one separately. And megakaryocyte actually going to develop into platelets, one of the most important types of our blood cells. Now let's go by each one of these. First, let's talk about the red blood cells. So proerythroblast, it's the first recognizable cell derived from the colony-forming unit, the proerythroblast. Now, what comes next is basophilic erythroblast, or we can just call it early erythroblast. And this type is smaller than the proerythroblast, arises by mitotic division. We know the whole thing is mitotic division type. And the cytoplasm, as you can see here, the cytoplasm shows strong basophilia. And that's due to the large number of free ribosomes. It has a large number of free ribosomes inside here. And so we can just say it shows a strong basophilia. That's why we call it a basophilic erythroblast. We can just call it early erythroblast. Now what comes after is the intermediate erythroblast. And you see here there are two colors in here. Now it shows, the cytoplasm displays both eosinophilia and basophilia. Eosinophilia is due to the staining of hemoglobin. So hemoglobin start synthesized in the early erythroblast, and it shows here in the intermediate the staining. We call it eosinophilia. And there is also the basophilia, which is also due to the staining of ribosomes. You know, ribosomes are important in the process of synthesis. We synthesize many things because we know the erythroblast, the erythrocyte, when it gets mature, it doesn't have a nucleus, so it doesn't synthesize anything. Hemoglobin is synthesized in the early stages of the maturation of erythrocyte. Okay, we will get late erythroblast, and then we get the reticulocyte. Now, before we get the reticulocyte in here, let me show you. This is reticulocyte. Before we get that, the nucleus almost disappeared. Reticulocytes, it's then going to become a mature erythrocyte. And if we noticed, the erythrocytes, the reticulocyte, actually they are about 5% of the blood of the erythrocytes in our blood. We have some amount of reticulocytes not only in the bone marrow, but also when erythrocytes going to the bloodstream. Let me write that down. Bloodstream, when they get mature, there are some amount of reticulocyte going with the. Now the other type of blood cell that the stem cell going to develop into is the myeloblast. Okay, now myeloblast is the other one other than the proerythroblast that will give us red blood cells at the end as the end product. The mature one that we have it in our blood. Now At the end of this pathway from myoblast, we'll get three main cells: basophil, eosinophil, and neutrophil. Now we know that neutrophil is about 60% of our leukocytes, and what I mean by leukocytes is the white blood cells. And we call these three cells as the granulocytes of white blood cells. Let me show you in another color. It's a granulocyte type of white blood cells. We have agranulocyte and granulocytes. Agranulocytes, we will talk about them in this pathway later on. But for now, let's focus on this one. This one starts with a promyelocyte or progranulocyte, and then we will get the myelocyte and metamyelocyte. Myelocyte is not showing here. Let me show you another one. So we see here, this is the myeloblast. We will get the promyelocyte, and then from the promyelocyte we will get the myelocyte. Now we have the neutrophilic myelocyte, we have the eosinophilic myelocyte, and basophilic myelocyte. From the myelocyte we will get the metamyelocyte. We have the neutrophilic metamyelocyte. I don't know if you can see it here. Neutrophilic metamyelocyte, eosinophilic metamyelocyte, and basophilic metamyelocyte. Now from this point, both basophils and eosinophils directly get to their mature form, eosinophil and basophil. For the neutrophil, first it gets immature form, we call it the bound neutrophil, and then to the mature form we call it a neutrophil. So we just have to skip this point in both of these types of cells. They don't have bound type of cell, they directly get the mature one. But in neutrophil we have the bound neutrophil. I just wanted to show that. And here what we talked about in the erythrocytes, we said we start as a pro-erythroblast, basophilic erythroblast, polychromatophilic. We don't need to get through each one of them, we just need to know that there is a reticulocyte, there is erythrocytes, pro-erythroblast, and so on. Here is the granulocyte type. We still didn't talk about the granulocyte of the leukocytes. We said the granulocytes, once we have the neutrophil, which is the major one in the leukocytes, 60-70%. We have the eosinophil, which is less than neutrophil in the leukocytes. And we have the basophil, which is much less than those. And if we look at the neutrophil, it's multi-looped cell, whether on the other hand we have the eosinophil, which is almost like it has two loops connected by some kind of chromatin thread or something. Basophil, on the other hand, is kind of different. It has many granules, as we can see here. We can't even see the nucleus. It has many granules. And yeah, those are the granulocytes. So if we come back to this one, lymphoblast, or we get at the end lymphocyte, and from the monoblast we get the monocyte. Monocyte then goes into the connective tissue and become macrophage. Okay, so lymphocyte and monocytes are a type of agranulocyte of white blood cells. Let's see them in more details. We can see here, this is the lymphoid stem cell. From it we get the lymphoblast, get divided into B lymphocyte, T lymphocyte, and natural killer cell. This is the agranulocyte type of white blood cells. And B and T lymphocytes, you can see the nucleus is almost filling the cell. The cytoplasm is very small portion of the cell. We have the natural killer cell. This is, as we said, the agranulocytes. There is no granules as much as in this type of granulocytes as passive eosinophil neutrophil. And we also see the difference between the nucleus. Here is mostly multi-looped, and in here it's just one big nucleus filling the whole. Let's get to the last type of cell that the stem cell develop into, which is the megakaryocyte. Megakaryocyte is the last one. Start with megakaryoblast, become a megakaryocyte, and then we will get the platelets. Now let's see how we get the platelets from megakaryocyte. And this also happened in the peripheral cytoplasm of the megakaryocyte. This is the peripheral cytoplasm. It can be divided into small compartments by invaginations of the plasma membrane. In these invaginations, they called platelet demarcation channels. Okay, in here in the plasma membrane, we will get the platelet demarcation channels. And the portion of the cytoplasm defined by adjacent channels, by these channels, this portion of the cytoplasm, for example, this will be a platelet that will be released from the cell. You'll see at the end will become a platelet. Okay, so we will have channels in here, and the cytoplasm, or the portion of the cytoplasm defined by adjacent channels, this will be platelet that will be released from the cell.